practice. In Jesus' name we have prayed. Amen. Okay, so I see we are 7 persons here, but I see that our presenter is already here. So I would strongly suggest that we go ahead and start. Except someone else has a contrary opinion, you may tell us if you think we should hold on a little while for others to join. But I think it's already 9 o'clock, we can start. So if there's another opinion, please let me know before I go ahead to introduce our presenter for today. Okay, so I believe the general opinion is that we should commend. Okay. So our presenter for today, for this topic, strategies for improving. Could we all please mute our microphones so we don't interrupt the meeting, please? Except you are to speak, please don't unmute yourself. Thank you. Okay, so our topic for today is strategies for improving vaccine uptake, addressing hesitancy and optimizing vaccination coverage. And this will be presented by a revered professor, Professor Ebelebi Onuibu, who holds a bachelor's degree in pharmacy, master's degree in pharmaceutical microbiology, and a PhD. She also holds a certificate in vaccines and viral immunology from the Harvard Medical School, USA, another certification on safety molecular pathology laboratory. Her research interests are vaccine development, viral immunology, host-pathogen interactions, bacteriophages, design, formulation, and delivery of drugs, especially vaccines, and her areas of specialty. Oh my God. Could you all please?
Moderator, you have to unmute yourself.
Thank you for drawing my attention to that. I didn't realize that I had put myself on mute. Thank you. So in continuing with the citation, her areas of specialties are nanomedicine, design, formulation, and delivery of vaccines. She's also a visiting professor or a visiting researcher, and a visiting professor to numerous schools in Nigeria and abroad. She has supervised many postgraduate students and has received numerous national and international grants and awards. She's also the editor-in-chief, Journal of Biologics, Research and Development, and has about 50 publications to her name, as well, and also has made contributions in 5 books. So you're very welcome to this special edition, and we are honored to have you present this topic to us, Professor Ebele Onuibu. You're welcome, Prof. You may have the floor, please. And sorry, before she goes on, I would like to please emphasize that we all allow microphones to stay mute so we don't interrupt the presentation. The presentation will last for about 30 to 40 minutes, and then we're going to have about 20 minutes to ask questions or make contributions. If you have questions, please jot them down. Just note them down somewhere. And then during the time for questions or contributions, all you need to do is just to raise your hand. You'll be acknowledged, and then you have the floor to make your presentation or questions. Thank you. Prof. Ma, you may please have the floor. Thank you.
All right. Thank you very much, Dr. Ihana Cho.
Very welcome.
I will need to, and good evening, everybody, to this edition of the CPAN, National CPAN meeting. Please call my attention if my voice is not audible. I will need to share my slides. I hope I can share it. Okay. Would the hosts be able to allow me to share?
Prof, you are a co-host, so I think you can share.
Okay. All right. Mm. Can you see my slides?
No, ma.
Okay, well, I don't know why it's not coming on. I've tried to. Let me try image from files.
But presently, Prof is not a co-host now, so you might not be able to see her slides.
Ah,
No, everyone is enabled to share the screen. We are seeing Prof's WhatsApp screen on the phone when she was sharing earlier. She's not about sharing ability.
Okay.
Prof, do you want to share the slide with the host or co-host to help you to share?
well, if former Moma has my slides, maybe she can share it. I don't know why I'm thrown off every minute.
It's not a co-host. Let me send it to the moderator, Dr. Ihanacho. Dr. Ihanacho, can I send it to you? Or Dr. Isa, who do I send to, please? Who can I send to, please?
Or if you send to me.
Okay. Give me a minute. Dr. Isa, I've sent to you. I've sent it.
Prof, you can go ahead now.
Okay. Is it showing now? Because
Very
Yes,
visible
it
now.
is.
Yes, it's showing.
okay,
It's showing now.
but I can't see it myself.
Oh. Oh.
Prof, you have to
Oh, okay.
close the WhatsApp participant list. That place that has participants
Sorry?
where you're trying to reopen your Zoom app from where you're trying to share earlier. You'll be able to see the screen.
That device. Okay, I can see it now. I can see it now. Okay, I'm sorry. I'm sorry for all the hurdles and obstacles. So my topic is actually strategies for improving vaccine uptake, addressing hesitancy, and optimizing vaccination coverage. My name is Professor Bele Onwibo. I'm a lecturer in the Department of Pharmaceutical Microbiology and Biotechnology, Faculty of Pharmaceutical Science, University of Nigeria, and Nsukka. And I want to talk about vaccines and how we can improve the vaccine uptake and coverage. So these are my outline, the topics I'm going to quickly go through. The introduction, that's the background, vaccination, types of vaccines, vaccine safety, vaccine hesitancy outcomes and addressing the hesitancy, then optimizing vaccine coverage. So it's very well known to all of us that we all get sick at one time or other in our lives, from infancy, continuing through old age. And these infections we know come from microorganisms, pathogens, because we have some microorganisms that are beneficial and they don't harm us. In fact, the percentage of harmful organisms as a pathogens are quite, when you compare them to the other pathogens, the other microorganisms, they are quite in the minority. But they cause diseases and they make us sick. And the infectious ones are spread from one person to another. And if you look at those pictures here, we can see people that are sick, like the hand that you can see at the upper part of the slide. It's someone with smallpox. Sorry, with monkeypox. That's the monkeypox infection. And then you can see someone sneezing with a cold. And then below, it's someone that has Ebola, the hemorrhagic Ebola. So some infections that come are very minor. They pass with time, while some are really, really deadly, like the Ebola virus is very deadly. And how does this microorganisms go from being an infection or causing an infection to causing a disease? Well, we all know the route of a bacteria or a virus causing a disease. There has to be an attachment. So the microorganism attaches, and then once it attaches and it enters the system, it can, if it's a virus, it will take over the machinery, the cell machinery, and start replicating and producing its own proteins, nucleic acid, and assembling them. Of course, by the time it assembles them and bursts out of the cell, it kills the cell, and then it infects another cell. And of course, some of the cells that they infect, especially these viruses, some of the cells that they infect are the cells that are even immune cells. So they weaken us more and more, like the HIV. We know that the HIV virus infects the CD4 cells, and the CD4 cells are like the major immune cells we have in our system. They control and regulate other cells. So the immune system is actually a network of cells, tissues, and organs that work together to defend the body against attacks by foreign invaders. So the body actually mounts a surveillance, also mounts a war against these foreigners, these pathogens, to prevent the host from becoming sick. But then if the person actually goes from infection, because infection is actually to attach, but then if the immune system overcomes the invader, the person gets better. after a while, the illness goes away, and then the person is well again. But then what would make someone that has an infection to go from an infection to actually having a disease? It's because the immune system has been overwhelmed. It's also because the organism is virulent, and it's also because of the population of the microorganism. So these three factors contribute to a microorganism actually gaining an upper hand over the immune cells. In this next slide, we will talk about the immunity, the body's immunity. We actually have what we call the B cells and the T cells that make up the acquired immunity. We also have the innate immunity. So these two arms of the immunity help us to actually stay strong, stay healthy. But then we actually introduce vaccines because just allowing the body to go through that process of having the infection and recovering is very risky, because we know that some people don't come out of it. The body is not able to put up a strong defense against the infection, and eventually the person dies from the serious illness. So we can even look at these pictures and we can see that nobody wants to have, to go through this debilitating kind of illness. It's actually a scourge. If we look at the picture here, we'll see someone that has smallpox, one of the most deadly, most deadly diseases, we've ever had in human history. So that's what the vaccine is here to prevent. We know that the vaccine is like a dummy of the real thing. So instead of someone actually passing through the trauma, of having the infection, coming out with this kind of symptoms, the vaccine mimics it, stimulates immune cells that would actually be released. And then, of course, what they are going to meet is a dummy, but then it has been released because they are seeing similar sequences, because what the immune cells actually see, not with eyes anyway, but what they actually, I will use the word see, what they see are sequences, protein residues, amino acid sequences. So once they see it, they already recognize the sequence as foreign, because that's one thing about the immune system. The immune system is able to differentiate between self and non-self. This is not me, and then it sees it as a foreigner and tries to attack. So even if it's a dummy, the immune system already sees it as foreign and already stimulates the necessary immune cells that would overcome the pathogen or the foreign body, either through phagocytosis or, well, there are many ways. Neutralization. Neutralization is actually that the antibodies that are released would coat the microorganism or the pathogen or the protein, whatever it is, the peptide, coat it and then present it to the phagocytes to phagocytose, or present it to the complement to fix it, or present it to the natural killer cells to kill it, or majorly this ways of actually overcoming the pathogens. So over time, since the time of Edward Jenner, we have generated vaccines. from the traditional vaccines up to the present time when we are now using synthetic biology, immunoinformatics to actually predict the right epitopes or the right proteins that would make very good vaccines. So each new vaccine presents several challenges. That is what vaccinologists face, because you have to determine the right antigen. Remember that the antigen comes from the pathogen, and the immune cells have to recognize the antigenic sequence. They are called epitopes. So they are called epitopes or antigenic determinants. And these antigenic determinants are sequences. It could be the nucleic acids, it could be the proteins, it could be the polysaccharides of the pathogen, but there is something that it has to recognize. And in this pathogen, they call them it's like a molecular pattern. They call the pathogen associated molecular patterns. So these are sequences that the antibodies or the immune cells recognize and then try to either phagocytose it or kill it with a natural killer, or the T cells have to come in. So over time, it has been the traditional vaccines, which have taken, initially it took many years, more than 10 years, to actually develop a particular vaccine, because you have to use the whole pathogen. And then, the whole pathogen involves actually growing it. So you can imagine having to grow an organism, whether it's a virus or a bacteria or a bacterium. You have to grow it to the level that would be enough to go around the whole world. It was a huge task, and it was expensive. So over time, newer technologies have helped us to, create better vaccines that overcome the initial hurdles that was involved in making vaccines. And then, of course, equally, bringing up the level and the number of vaccines that are able to be distributed all over the world. So now we have the immunoinformatics, the reverse vaccinology. Of course, reverse vaccinology involves just going to the computer, clicking, and then the algorithms come out and show you the right proteins that will give you the right the right vaccine. So it was no longer the whole thing of going back to grow the organism, then identify the antigenic determinants, and then purify it, and then formulate it into a vaccine, and then with all the phase 1, phase 2, phase 3 clinical trials of making sure that it is safe, of making sure that it is efficacious, if the efficacy is almost 100%, and then making sure that the dose is right. So, but now with immunoinformatics, it's just a click away, because after the Human Genome Project, many other organisms have been banked. It's now in NCBI. Just go to the gene bank, and then Ebola virus sequences, genomic sequences, or these other viral genomic sequences, and then you throw it in, and then it comes out showing you. And then it's now all about synthetic genes. All these bioinformatics companies or biotechnology companies, they make synthetic genes. So they go from the synthetic genes to formulating the vaccines. So a vaccine that used to take up to 10 years to produce now take less than a year. And that was actually what happened in the one of the COVID-19 vaccine. So we could ask ourselves, why do we have to go for a vaccine when we have drugs? Well, there are many reasons that one can give. It's not easy to get drugs to everyone. And then most importantly, to give a drug, you have to diagnose, you have to run tests, you have to diagnose, and then you treat. By that time, the disease may have spread, but we know the benefit of vaccines. It prevents. You don't have to see the person actually come down with the disease before you start treating. So vaccination, and what is actually vaccination? Well, vaccination is actually to introduce a biological organism, which can be in the form of the whole organism or a part of the organism into the body to generate immune cells. And I wanted to talk about what microorganisms actually do to the body. You know, the first thing is, for those of us that are in the lab, when you grow microorganisms, you on cells, what you're going to see to really show you that the virus, let me use the virus, what will help you know that the virus is actually does exist is that you're going to see plaques. You're going to see black spots. So that those black spots show that the virus is growing and then it's killing the cells that is growing on. So that is what actually happens in the body. We call it the cytopathic effect. So viruses cause cytopathic effect. It grows and kills the cells. And once it kills one cell, it moves to another cell and kills it, destroys it, lyses it, comes out with the progeny, and then they move to another cell, kill it. So you see infected cells that have already died. So that's one thing. Another thing that viruses can do is that they can cause cells to become cancerous. They can transform cells and they become cancerous. So it's not just infection, but the cell now becomes cancerous and also dies. Or it could also induce, viruses, for example, can induce the immune system to react with an inflammation. And those inflammations can also be deadly. We remember what happened in the COVID-19 period, pandemic, where we're talking about the cytokine storm. So it induces the immune cells to actually release inflammatory cells that are so bad that the person actually dies from the inflammatory reaction, much even more than the virus itself producing those cytopathic effects. So these are three ways that actually viruses can cause the death of the host. So, but if we already have vaccines for all these emerging viruses, deadly viruses, then we're protected because the body already has the antibodies, neutralizing antibodies that are there. The memory cells are there, memory B and T cells are there because they've been exposed to a dummy. So they're already mounting surveillance for any opportunity of meeting the real pathogen. And then not just the B and the T cells. Of course, we also have the innate cells, the innate immune system that also releases interferons. Interferon alpha and gamma are interferon alpha and gamma cells help. There is noise in the background. So the interferon alpha and gamma cells help in the innate immune system. They help to actually inactivate the pathogen from actually attaching, because the pathogen always looks for its receptors. It has its own receptors, and it looks for the receptors on the hosts. And sometimes some pathogens have many receptors that they can attach to and cause the disease. So with all these stimulated immune cells, the body is protected. So we can actually gain a lot from vaccinating ourselves, rather than allowing the natural infection to run its course, because it could actually be deadly. One may not survive it. So we have the different vaccine types. We have the live attenuated vaccines, the inactivated vaccines, subunit vaccines, toxoid vaccines, the conjugate vaccines, DNA, and recombinant vector vaccines. So if you permit me, we could look at them briefly. So the live attenuated vaccine. Well, a live vaccine is actually a pathogen, either a virus or a bacterium, that is mutagenized in a way that blocks its ability to cause serious disease. There are many ways to mutagenize a microorganism. It can passage it through many media that is not the natural host. For instance, a human virus that is passaged in a chicken will come out mutagenized. It will be too weak to cause a disease again, but it has to be passaged many times, up to 20 times. With the advanced technology, you can also mutagenize by actually going to the gene itself and changing, knocking out some genes, mutating the genes so that they don't become virulent anymore. But they will still be alive, because that live virus that actually makes them mimic almost 100% what happens in a natural infection. So the microorganism actually grows, multiply, replicates, but doesn't cause any disease. But the immune cells don't know that it won't cause a disease, and will still stimulate memory B cells and T cells to actually to destroy the pathogen. So the good thing about the live attenuated vaccine is that it induces memory cells, just like the wild type infection. But it shouldn't be given to immunosuppressed people, because it could replicate in an uncontrolled manner and cause the actual disease. And we have some examples of the live attenuated vaccine. We have the MMR, the yellow fever oral polio vaccine, BCG, and the Ty21A for typhoid. So we have the inactivated vaccine. So these inactivated vaccines are the pathogens that have been killed by heat or chemicals. Use formalin to kill the pathogen. But you have to verify that it has to be killed in such a way that the organism is killed, but the antigenic determinant is not tampered with, because the organism has to actually induce the stimulation of immune cells. So, like we do in the lab, you have to keep checking the time and the temperature, increasing the temperature gradually and checking the time until you get to the end time or end point where this is the time at which it kills the organism and at this temperature. But it doesn't destroy the folding of the proteins. So the proteins are still in their natural state. It's not destroyed, because once you destroy the proteins, it's actually the proteins that stimulate the strongest immunity. Actually stimulates the germinal centers of the immune cells to actually mount a strong immunity against the microorganism. So it kills the organism, but it doesn't destroy the antigenic determinant. So we have some of the kill vaccines, like the hepatitis A, some flu vaccines, the Salk inactivated polio vaccine. Then the next one is the subunit vaccine. So these subunit vaccines came like second generation of vaccines. The first generation of vaccines were the live attenuated, the inactivated, the toxoids. So these subunit vaccines, would call them the second generation of vaccines, where vaccinologists decided to do away with the whole organisms. After all, we don't need the whole organism. We just need the proteins that are going to stimulate the immune cells. So through different methods, they break open the organism, lyse it, release the proteins that are needed. It could be by centrifuging the organism at very high speed, very, very high speed, and you break it open and take the parts, the proteins that are needed, multiples of them, and then formulate your vaccine with it. But then the thing about this inactivated and subunit vaccines is that you need to give the injection more than once for its durability. They are not very durable, so you may have to give booster doses or
I think the presenter's network is a little bad. Let's hold on a little for her to come back, please. Thank you.
Okay, I was muted, sorry. So the subunit vaccines, like I said, they require boosters, they require adjuvants to be able to stimulate a good response from the body, from the immune cells. Then, so we have the examples, the hepatitis B surface antigen vaccine, which is also recombinant, then the human papillomavirus vaccine, the acellular pertussis influenza vaccine. Then the toxoid vaccines are also extracted from the whole organism, because this is mostly from bacteria. We have Corynebacterium diphtheria, we have Bordetella pertussis, we have Clostridium tetani. All these are made for toxoid vaccine. So the toxin is extracted and modified by heat or chemicals so that it's no longer dangerous. And that is actually the target of the immune cells, these toxins that are the exotoxins. These are, I mean, in microbiology, we call them the super antigens from this very big microorganisms. So we make toxoids from it. So we have the DTaP and the Tdap from diphtheria, tetanus, and their cellular pertussis. Then we have the conjugate vaccines. So conjugate vaccines also came straight after the subunit vaccines. And what happened is that the conjugate vaccines are actually coming from polysaccharides from bacteria. Some bacteria produce capsules and they are polysaccharides. So, and they're very antigenic, very, very virulent and antigenic. But then the immune system of children do not recognize these sugars. So they don't produce robust memory cells. So the thing is that it's conjugated, is actually linked to proteins because proteins are very good in inducing the T cells. So polysaccharides are T, we call them the T-independent, like antigens. They don't stimulate T cells. But when you link them to proteins that are T-dependent, that's T meaning T cells, they are recognized. And then the immune cells recognize the proteins, are released, and then because they are linked to polysaccharides, they also become, they also react to the polysaccharide, thereby mounting an immune response to the sugars, to the polysaccharides. So we have Neisseria meningitidis, meningococcus vaccine, the pneumococcus vaccine, the HIB from Haemophilus influenza type B vaccine. So they are quite good. And now they have many before, and some of these bacteria have different strains, very many strains. Some have up to 23 valencies. I mean, so with time, they are increasing because the different strains react differently to the vaccine. So as many of the strains are able to get, to have a vaccine for the better for the patients. So the next is the nucleic acid vaccines. The nucleic acid vaccines, that's the RNA and DNA vaccines. Well, we saw for the first time the RNA vaccines during the COVID-19 pandemic. It was actually produced. The DNA vaccines are still in clinical trials. So what happens in this RNA vaccine is that they are nucleic acids. So originally, they are actually cut out, and is a process of actually cutting out the DNA vaccine. So they're called target genes. So these target genes is because it has been studied. So these genes produce these proteins, and these proteins. So for example, the SARS-CoV-2 is an RNA vaccine, is an RNA virus, and it has spikes. Those spikes are on the envelope and they are proteins, but then they have genes that encode them. So it's actually to go to the gene, cut out, splice out the gene that encodes these proteins, because it's the proteins that the immune cells, the T cells will react to. So it's spliced out, using endonucleases, and then is inserted in a carrier. It could be inserted in adenovirus that has been mutated. So there's a protein there called the E1 that is mutated so that it doesn't replicate. And then adenovirus is big, so you can actually insert it therein. So the same endonuclease splits open at particular sequences. We call them palindromic sequence. So at a particular sequence that is recognized, it will cut it and insert your gene of interest. And then you ligate it. You ligate it again, and then it's a lot of trial. So after ligation, you now watch to see if the proteins will be expressed. So if the proteins are expressed, of course, through chromatography, through, what's the word again? You can actually ascertain whether the protein is electrophoresis. You can actually ascertain whether the protein has been expressed. And so what the idea is in this RNA vaccine is that the body becomes a machine, for producing this protein. So it's like the immune cells keep meeting those proteins that are being expressed because the adenovirus keeps expressing them, just like it's expressing its own proteins, and just like the host cell is also expressing its own proteins. So these proteins, these spike proteins are being expressed, and the immune cells recognize these sequences and then keep releasing immune cells. So the body is forever releasing immune cells against these proteins and cause the prevention of the host coming down with the disease if a real live pathogen is encountered. The DNA vaccine is also like that, but it's still in clinical trials. Because the good thing about the RNA vaccine is that it doesn't go into the nucleic acid. It's an RNA. RNAs just translate. If it's a messenger RNA, it translates into proteins, and it does this in the cytosol, in the cytoplasm. So we have, last of all, we have the recombinant vector vaccine. So it's still the same whole idea, of using genetic engineering in producing the vaccines. And one thing with the engineering is that the messenger RNA of viruses are different from the messenger RNA of human hosts. So the human host, the messenger RNA, we call it capping. It has a 5' end and it has a 3' end. So in the 5' end is capped. We call it capping. So it's capped with the 7-methylguanosine, and then there is a ribose there. The second carbon has a hydroxide, and there's also another cap. We call it a methylated cap. So, and also at the 3' end is also capped. So it's capped. But then the virus doesn't have these caps. So that's how the body recognizes the differences and destroys the RNA. But then with this genetic engineering, they're able to, before they insert it, they cap the messenger RNA of the virus so that the host does not recognize it. Because if the host recognizes those double-stranded RNAs, it will destroy it. Endosomally, they have things that are stimulated that would immediately recognize the difference and quickly destroy it. So that way, the body, so the adenovirus is inserted in the body, and then the adenovirus expresses the proteins and the immune cell responds by producing antibodies and T cells. So with this recombinant vector vaccines, we've produced Ebola and several COVID-19 vaccines. So we come to vaccine safety. And it's actually normal, it's actually understandable that people are concerned about the vaccines they take, the safety, especially since the COVID-19, which was produced very fast. There was a lot of hesitancy, there was a lot of panic, more panic than the disease itself, about the benefit of the vaccine they are taking, what's the aftermath, what's the long-term effect, what's the short-term effect. Well, yes, we're humans and we can make mistakes, but then we should also know that it's not one person that is involved. There are a lot of regulatory bodies that are involved in making sure that the vaccines that are produced are standard and they are faithful to the, they are faithful to the protocols that have been approved. Well, in the past, in 1955, there was a vaccine failure of the polio vaccine. It was inactivated vaccine that was actually contaminated by a polio virus that had not been inactivated. So after the inactivation, it was not done well. So there were still some unkilled the virus that contaminated the batches, and up to 40,000 to 70,000 cases of polio were documented, and up to 200 children actually were paralyzed. But after that, more surveillance and more safety measures were actually put in place to make sure that it doesn't occur again. Then there was also another incident of the same polio vaccine, the inactivated vaccine, between 1955 and 1963, with a contamination with the SV40 virus. SV40 virus is a virus that transforms cells to look like cancer cells. So the culture cells, the cell culture that they were growing the polio vaccine, they were growing the polio virus in, was contaminated with this SV40. And this got into the vials and actually caused a problem, to the recipients. Then another one was also another contamination around 2010 of rotavirus vaccine that was contaminated with the porcine sickle virus. But this porcine virus actually affects pigs, but not humans. It doesn't cause any disease in humans, but still, it was still something to be noted and worked on, be more careful to make sure that it doesn't repeat again. Then another time, way back in 1976, there was also the swine flu vaccine that was associated with the Guillain-Barré syndrome. But then, so this Guillain-Barré syndrome is a peripheral autoimmune neuropathy that causes demyelination and paralysis and eventually cause death. But then that flu vaccine, it was just 1 in a million people that developed the syndrome. And if you compare it to those that had the swine flu itself, if you compare it to people that had the Zika virus or people that had Campylobacter jejuni infection or cytomegalovirus, if you look at the upper part of the slide, we could see that it was very low compared to, because other diseases cause this syndrome. So to actually stick or attach a stigma to the swine flu vaccine that is causing Guillain-Barré syndrome, which compared to those that had Zika virus and those that also had Campylobacter jejuni infection, it was actually insignificant. It was very low, and shouldn't actually cause a hesitancy in taking the vaccine. Then there was also the hepatitis B vaccine that was associated with multiple sclerosis. That was in 1998, but it was never established. It was just a fraudulent news that was never established after investigations were held. Then another time in 2009, 2010, another flu vaccine came out, the H1N1 flu vaccine, and people in Finland found out that it was associated with narcolepsy. Some people were falling asleep spontaneously, and it was investigated, and it was discovered that there were not many people that it happened to, and then it didn't continue. It was not a problem that continued afterwards or in other countries. Then we now go to the one that is currently going on now, autism. I guess that everybody is actually in on the news that vaccines cause autism. In fact, I took this information from the Google newspaper, where on November 19th, the CDC webpage that previously stated that there was no link between vaccines and autism has now been updated, and it now reads that the claim vaccines do not cause autism is not evidence-based because studies have not ruled out the possibility that infant vaccines cause autism. So now the studies supporting a link have been ignored by health authorities. The Department of Health and Human Services has launched a comprehensive assessment of the cause of autism, including investigations on plausible biologic mechanisms and potential causal links. So we know that this is actually political, and there's a lot behind it. So there is no scientific proof. There is no scientific proof that vaccines cause autism. I've met a lot of people that have told me that their children are autistic because of the vaccines that their children took. I know a woman that has two boys. Well, they are men now. And two of them, the two sons she has, are autistic. And she always refers to the vaccine that the children took. But there is no correlation. There's no correlation. And correlation does not mean causation. Even if it happened, like somebody might have a vaccine immunization and then probably dies some days after, and then it's claimed that it was a vaccine the person took, that it could be something else that caused it, not the vaccine, or maybe there were other underlying illnesses. So as of August 2024, approximately 13.72 billion doses of COVID-19 vaccines have been administered worldwide. The deaths were not above 0.0017%, which is extremely low. And if you compare it to the number of deaths of those that had the COVID, then this is actually insignificant. And the vaccine up to today is still being monitored. It still passes through the clinical trials, and no vaccine with a clinical trial of less than 75% is allowed to be issued now. It has to be as high as 80-something or 90-something to really show its efficacy and its safety. So there was also another particular safety issue, the thrombotic thrombocytopenia. So this rare type of blood clot was linked primarily to the adenoviral vector vaccine of J&J and AstraZeneca. But it was a fake news, and there is no proof of the linkage between the vaccine and the syndrome. And I did also one of the myocarditis, but this one of the myocarditis, it's actually, for you to actually establish that it was actually caused by a vaccine, there has to be a biopsy that especially, diagnoses the illness, that this is actually a vaccine. Because in the biopsy, you have to have traces of the vaccine there to really show that yes, it's actually the cause of the myocarditis. Then anaphylaxis, yes, many people have allergic reactions to drugs, to vaccines, to food, to anything that has protein. For some people, it causes an allergic reaction, and it has to be checked. Someone has to know herself and her reactions to actually. But then it can be controlled by giving epinephrine, to the patient if there is such a reaction. But then the reported cases were as low as 2 to 10 cases per million doses given. So the vaccine hesitancy, now, after having heard a lot about the vaccines we have, how the work, the mechanism of action, we'll be able to educate our peers, we'll be able to educate other healthcare workers, we'll be able to educate relatives, we'll be able to educate the community on the benefits and the safety of vaccines, and also that no individual produces a vaccine and releases it. It has to pass through a lot of stages. It has to be given to volunteers from different parts of the world, different races, different genetic makeups, to actually see both the efficacy and the safety of that vaccine. Because actually, the issue is actually with childhood vaccinations. A lot of people are saying that the number of vaccinations for children have increased so much, and then with the increase in the vaccinations also goes with a surge in autism. Some people claimed also that is not just the vaccine, the organism that is causing the syndromes that they are experiencing, but also the excipients. There is this claim from CDC or HHS, that's the Health and Human Services of the US, that is actually the aluminum that is the adjuvant that is included in these vaccines, that is actually the cause of this autism. But there is no cause, there is no link, between the two. So the drivers of this vaccine hesitancy are actually misinformation, lack of communication, lack of knowledge, cultural and religious beliefs, distrust in government and pharmaceutical companies. People believe that the government has been bought. Pharmaceutical companies are trying to make millions from the people, and they're the ones, we hear all sorts of things, that they are the ones, laboratories have released this viruses so that pharmaceutical companies can make money from them. And then it could also be some personal experiences, especially if someone has reacted badly to a vaccine, an immunization. Nobody else wants to get vaccinated because of that allergic reaction. So a lot of influences, social media, peer pressure, socioeconomic factors, all these contribute to the vaccine hesitance. Sorry, I have to move because I've taken quite a lot of time. So we can try to.
Hello, Prof.
Yes.
Sorry, I have to interrupt. I'm glad you've noticed the time. I think if possible, you could round up in 5 minutes, if possible.
Okay. So
all right, thank you, ma'am.
Yes. So all this misinformation has actually led to increase in diseases, pockets of diseases in different places. We must have heard about it. Some parts of the world where because of the distrust, they don't want to vaccinate, and the children are coming down with measles and other, other types of diseases. There are now higher hospitalization rates, and it's not just one individual that gets infected. If one person gets infected, we know that the thing will spread to other people, and then that would actually lead to increase in death. So how do we address this vaccine hesitancy? Well, we have to tell our stories. There is someone that said that he works in a health facility and said he's a pediatrician, and when he started his profession as a pediatric doctor, they used to see a lot of children die from all this infection. But with these conjugate vaccines, they no longer have these diseases caused by pneumococcus that used to terrorize them and kill so many children. So people have to tell their own stories. We've seen the pictures of people with small Ebola virus, monkeypox. Is this what we want to go through? And who knows, one may not survive it. So we have to tell personal stories so that people realize that the implications of listening to false information from media campaigns and the rest of them. So I think I'll stop here. These are my references, and thank you very much.
Thank you very much, Prof. We really appreciate your time, the efforts you put in. This was quite a very deeply researched presentation. We thank you for the time you've given us. Once again, everyone, I'd like to let us know that this presentation was done by Professor Ebele Onuibo, a pharmacist and a PhD holder specializing in vaccine development. Okay, with research interest in vaccine development, viral immunology, host pathogen interaction, and a whole lot under that area. And a lecturer and a visiting researcher in Nigeria and abroad. So we are grateful that you gave us this time. You shared your insights with us and helped us to have better understanding of this. So at this time, I would like to open the floor for questions or comments. Please, we already have our time so far spent. I don't know if 5 minutes would be okay for us, but maybe we could just work with that 5 minutes. But if you need more time, we could add to it. But for now, let's try to work with 5 minutes. So if you have questions or comments, could you indicate by raising your hand, and then you'll be duly acknowledged to have the floor. Anyone? I can't see any hand up. So I guess the presentation covered most areas in our minds that we don't have any area that appears ambiguous to us. Okay, I see one hand up. Dr. Felicia Williams, please go on with your questions or comments. Dr. Williams, please.
Yes. Good evening, everyone.
Good evening, ma'am.
Yes, we want to appreciate Professor Ebele Onuibo. The area of concern has to do with clinical trials with these vaccines. Yes, some clinical trials must have been conducted outside the shores of Nigeria. What are we doing to ensure clinical trials are conducted on these vaccines, or even post-market surveillance on these vaccines? Thank you.
Sorry. Well, are you talking about clinical trials in Nigeria or clinical trials abroad?
I said clinical trials may have been conducted on some of these vaccines abroad. Do we have any effort going on to ensure that clinical trials are also conducted locally in Nigeria? And apart from that, post-market surveillance on these vaccines? Thank you.
Well, I believe that clinical trials, once conducted and approved, there is no need to do extra clinical trial, because clinical trials actually take time to do and a lot of money. In fact, I think that the majority, the most part of the expenses government spends is on clinical trials. So once clinical trials have reached the stage 3 or 4, which involves thousands of people from different nationalities, and it's finally approved by FDA, CDC, and other regulatory bodies, then it's closed. Then post-market surveillance now starts. And they keep following it and recalling if there's any documentation of any adverse effect, and it's actually noticed by many people, then it's recalled. But I don't think once a vaccine is certified, you have to go back to do clinical trials again. My thoughts. Mm-hmm.
Okay, thank you, Dr. Williams, for that question. And we also appreciate Prof. Ebele for the response. I believe you're satisfied, Dr. Williams. Okay, there's another hand here. Salam Theowe. Please, you may go on with your comment or question. Pharmacist Theowe, please, you may go on with your question or your comment. I see your hand. Are you hearing me? Hello? We can hear you clearly.
To your last question, to your last presentation, and you said that most times, if I may be correct, it is the adjuvant, or it is the adjuvant that can cause side effects or adverse effects for people in the vaccine. So I wanted to know, why is it happening? Are they not aware?
Actually, I said that the claim, there's a claim that the alum that is added in as an adjuvant in one of those vaccines was tied to the autism of children. But it's a claim, there is no scientific proof.
So you're satisfied with the response, Pharmacist Theowe. I think we can move to the next person whose hand is up. Please, are you okay with the response, or you still need further clarification, Pharmacist Theowe? All right, so your hand is down now. So, Pharmacist Innocent Onoja, could you please go on with your question or your response or your comment?
Thank you. My comment is to appreciate Prof for lecture, and to request, because of the nature of the network, it was actually interrupting most of us, well, to me in particular. So I don't know if we can have access to the slides at the end of this lecture. Thank you. Did you get me?
Yes, it's available. I mean, I guess it can be made available to all of you, whoever wants it.
Thank you.
You're welcome.
All right, thank you, Pharmacist Onoja. I see Dr. Felicia Williams' hand up again. Please, ma, maybe 1 more minute, please. Thank you.
Yeah, I want us to look at the differences with regards to ethnicity of the different countries, and the initial population that were included in the clinical trials, initial clinical trials. It may not be very, very representative of other countries where this vaccine can be used. We know generally in Africa and in Nigeria, clinical trials issues, it is only of recent that it is being considered. So if we have opportunity to be involved it is good we raise this issue, especially where the clinical trial has been conducted in countries where FDA has given the approval. Then 2, for the post-marketing surveillance, I don't know how we are doing here in Nigeria, because NAFDAC was initially saying that individual organization should be doing the post-marketing surveillance. I'm of the opinion that for them to do that, it will really be, how will I put it, the authenticity, if it is being done by the individual organization, can really be questioned. I don't know whether you have any information with regards to that.
No, I don't. I don't actually have, because I'm actually not in public health. So I am in basic research. So though I read a lot about the underrepresentation of colored people in their clinical trials, I've read a lot about it, and it's something that actually should stimulate us, which we have been crying for years, for us to have our own vaccines, vaccines that are produced in Africa, because Africans produce less than 1% of the vaccines that are produced, in the world. We depend on the Western world, so with the COVID-19 vaccine, we have seen how much we're actually dependent on the Western countries. So for some of us that are well connected to the government, to the policymakers, we have to keep making our voice heard, that it's time that we started producing our vaccines so that the clinical trials can actually showcase, what we have locally, and actually bring in all the peculiarities, that we have in our own settings.
Okay. Yeah. All right. Thank you very much, Prof. So I think having established that we are very much dependent on the vaccines produced with so much underrepresentation of our own peculiarities like the blacks. So I think we should we should actually pay a lot of attention to raising awareness on the post-marketing surveillance of these products, particularly among facilities where these are given to help mitigate or prevent, either primarily or secondarily, any form of unwanted effects, or raise awareness of any reactions, just things that pertain to post-marketing surveillance to help ourselves. So we as pharmacists may just take that as a responsibility to always emphasize on that and raise awareness on that, pending when we will start having our own productions here in Africa or in Nigeria. So having exhausted
Is that the case? Eh? Eh? Okay. Why is it?
Oh, sorry, I didn't know I was mute. Okay, so I said I was thinking we are done, but I just saw another hand up. So Elias is if you could do with 1 minute rather, please have the floor. Thank you.
Okay, thank you so much. For Marcela Sokachikwe is my name from Baisa State. I just want to ask one important question about the issue of adverse effects following immunization. Do we really think that inappropriate vaccine storage can be contributing to adverse?
Hello?
I think
Hello?
his network is not bad. He's still on, but he can't be heard. Should be his network. Thomas and Elias, if you can hear us, we can't hear you anymore. Maybe you could send in your question as a text if you can't, if your network is really bad. Okay, so having lost him, I think we could go ahead and end the presentation for today. We appreciate everyone who has been available for this presentation from the beginning. We also appreciate those who joined along the line.
Hello?
Oh, okay.
Hello? Oh, network is
Yeah,
actually very bad here.
we can hear you now, please. You may go on.
Yes, I was asked 2 questions. It's actually one suggestion anyway. Do you think that the issue of improper storage of vaccine could be contributing to the issue of adverse effects following immunization? And the second question is the issue of giving of live vaccines to exposed infants, particularly children born of HIV-exposed mothers. We know that some of them can be exposed, can be infected actually, and the way you give them live vaccine can be contributing to the disease itself. I'm talking about the live vaccine, like the BCG that's given at birth and the rest of them. Do you think we can still go on and continue giving them live vaccine, knowing that they are exposed and they can actually be infected with HIV, thereby causing the disease you want to prevent if you give the vaccine? Thank you.
Okay, thank you very much. With respect to improper storage, well, vaccines are like drugs. The vaccine has not just the organism. Vaccines have been made like drugs with antibiotics to prevent contamination, and has adjuvants, has stabilizers, has antioxidants. So when there is improper storage, and when you say improper storage, what comes to my mind is lack of cold chain. Then a lot of chemical reactions could actually start taking place. So it's possible. Just imagine a drug that is not stored properly. What could happen? Apart from the fact that it's no longer potent. So the vaccines definitely will not be potent and will not have any effect. It's no longer efficacious. It could also cause, maybe oxidation could occur, hydrolytic, changes could occur. It could be from lights, maybe some vaccines that are meant to be stored in dark places where there is no light, and they are stored in the open where the light affects it. So there may be some chain reactions happening that could actually result to some adverse effects. I agree it could happen. And then live vaccines should not be given to immunocompromised patients, whether HIV or other kind of granulocytopenia, people that there are some people that don't even have, immune cells, good immune cells, so they have to be given antibodies, they have to be injected antibodies. So whatever is the case, should not be given live vaccines. So with these children, if they have HIV, then they shouldn't be. But if they don't have HIV, then I believe they can be given the live vaccines.
Why I'm asking is that, if you go, when we want to do when we want to give the vaccine at birth, of course, at that time, the infants are not diagnosed of HIV, even though they are given birth by those pregnant women. But we'll still give them BCG because it's supposed to be given at birth. Being a live vaccine, I'm looking at the risk it imposes, on the exposed children, particularly if they are infected with HIV.
Well, I don't know. I don't really know it doesn't change. Well, I think it's logical that they shouldn't be exposed, but there is no other way out of it. So maybe, I don't know. It's possible that they are not immunocompromised. I don't know if any other person has a better suggestion.
I think it's based on risk-benefit analysis. The
Yes.
child will be properly assessed. If the child is born of a mother who has HIV, then the child would definitely be thoroughly assessed after birth to see the level of, to see if the child is infected. Of course, you may not really notice immediately. You have to test again, and then of course other tests will be done to check the child's eligibility to take any of those vaccines. So it may not really be a one cap fits all. It will be based on individual child assessment. Okay, so I see 3 more hands, including our chairperson. So I'll give our chairperson the floor to speak. Then after she has spoken, pharmacist Theowe's hand is up again. Maybe he could speak a little, but let our chairman speak for now. You may have the
Thank
floor.
you so much. I would have loved to speak last, just for the closing remarks, and we'll end the presentation. Thank you. Hello? Can you hear me?
I heard you, ma. Thank you. Thank you,
Okay.
ma. I heard you. So I said pharmacist Theowe's hand is down now.
Okay, do I
Yes,
go
my
ahead?
hand. My hand is up, Mrs. Onoja.
Your question was just addressed now. So if pharmacist Theowe's hand is up, okay, you may go
Yes, no,
ahead.
I have it. I do. I typed it on the chat room, but I have it. My question is, or my concern is, issue of live vaccines and immunocompromised persons. So, what is the fate of people that are immunocompromised, especially HIV, persons living with HIV and the yellow fever vaccine? Yellow fever vaccine.
Okay. You well know that live vaccines are traditional vaccines, and right now little live vaccines are made because of the cost. So we're now going with the advanced technology. They are now subunit vaccines, recombinant subunit vaccines, the RNA vaccines, and so it mustn't be the live vaccine. Can give killed vaccine, can give subunits. So it mustn't be the live vaccine that is given to the patient. It's just a warning that if the person is immunocompromised, the risk is higher than the benefit.
All right, thank you. Thank you, Prof. I believe his question I believe he's satisfied with the response. Okay, so Pam Theowe's hand is up again. Pam Theowe, please, 30 seconds. Thank
My
you.
hand is are you getting me?
We can hear you.
Okay,
We can hear
so yes, I want to find out because most times vaccines, immunologists are associated with pharmacists. So I want to know what should pharmacists be doing at large scales if it comes to vaccine?
Sorry, I didn't get the question.
I said,
Dr. Yanacha, can you repeat it for me?
yes, are you getting me now, Claire?
It's clear now. It's clearer now.
Yes.
You may go ahead.
I said, sometimes I believe that vaccines, immunologists are associated with pharmacists. So I want to know what should pharmacists be doing at a larger scale when it comes to vaccine?
Okay, she's asking what pharmacists should be doing on a larger scale regarding vaccines. Since vaccines are usually associated with pharmacists, what are pharmacists' roles in vaccines? I think it's very broad. Is it in vaccine uptake, in vaccine dispensing, in vaccine production, in vaccine awareness?
Oh,
So approach from many
okay.
angles.
Well, yes, I would say that we're actually just like drugs. Vaccines are drugs, and vaccines are produced in pharmaceutical companies. So we're there in the production. We're there in the assaying, because it's part of the quality control. You have to assay what you have produced. So it's not just, oh, I've identified the antigen, then I make the vaccines. I have to run assays to really realize. So we're actually everywhere. It's a continuum where you start and you finish. But then you can't do it alone. You have to have other scientists as part of it. And pharmacists are also immunologists. They also are good in immunology. We're expected to know the response, just like a pharmacologist has to know what's the effect of the drug on the system and how does the body relate to the drug. What's the effect of the drug on the body, and how does the body, in terms of how does the body excrete, how does the body, the whole thing of, the pharmacokinetics and the rest of them. So vaccines are drugs. So we're there at the core as the key drivers of the production.
All right, thank you, Prof. So having exhausted all the questions and comments, I think I'll let our chairman have the floor so that we can close for the night. Thank you all that have contributed and asked questions. Thomas the Elias, I see your hand, but we are way behind time. So could you please just type in your question? I don't know. Okay, maybe 30 seconds, please. 30 seconds.
Okay, I have concern about the issue of vaccines in many hospitals because in most hospitals, vaccines are not controlled by pharmacists, even though we say they are drugs. So what can we do to have access, to have a custody of these vaccines in most of the hospitals? If you go to most hospitals, you see nurses having, custody of these vaccines. Is it that we don't have enough pharmacists in our country? Why do we still allow them to have access and custody of the vaccines, which are also drugs? Thank you.
All right. Thank you. Thank you, pharmacist. I see a comment here that will be beneficial to all of us regarding a question that was asked about immunosuppressed or children born to HIV mothers. Someone has sent in a write-up on WHO guideline for such conditions, and it reads, WHO guidelines for immunizing children with HIV infection and infants born to HIV-infected women differ only slightly from the general guideline. BCG and yellow fever vaccines should be withheld from symptomatic HIV-infected children. Only one serious complication has been attributed to measles vaccine administered to severely immunocompromised adults, and that is fatal pneumonia. Although 2 HIV-infected infants have developed vaccine-associated paralytic poliomyelitis, several million infected children have been vaccinated, and the evidence does not suggest that there is an increased risk. The benefits of measles and poliovirus vaccines far outweigh the potential risk in HIV-infected children. The policy of administering routine vaccines to all children, regardless of possible HIV exposure, has been very effective in obtaining high immunization coverage and control of preventable diseases. Thank you for the person who sent in this. So with that, I think we will give the floor to our chairman so we could round up for the night. Thank you.
Good evening, everyone.
Good evening.
I want to start by appreciating my dear friend, Professor Ebele Onimo, for this well-detailed presentation. Thank you so much, Prof, for taking
Thank you,
to
ma'am.
educate us today on vaccines. And also our dear moderator, Dr. Ihana, so thank you so much. During the course of this presentation, I cited the important stakeholders of my profession here. I saw Professor Bolajoko Aina, Dr. Fumilayo Oluwatoyin. Okay, also our dear Education Committee Chairman, former MOMA. I also want to appreciate the neck of CPAN, who we are here since, Dr. Issa, Dr. Diajulo, Pharmacist Fonta Ronald. Thank you so much for joining us. This topic tonight is very, very important for us pharmacists. Earlier, we pharmacists shy away from immunization and vaccination, but it has come that we must have to put our interest in this in terms of public health, public awareness, educating our patients, the public, in the community, for them to really understand that those myths are all lies, so that their children should be well immunized. And not only that, we must have to go into certification. Those in the community should go into certification so that they can start even immunization in their pharmacy. Yes, if you can get satisfied, you can do that. So let's only go into public health. It is very, very important. That's the only way we can reach the community, educate them, and take our stand, okay, on this topic. To create that awareness, educate the populace concerning immunization. There are so many myths because you see some parents, they won't bring out their children for immunization because of what they heard, which may be all lies. So it is our role as pharmacists to really educate our communities. Let us always move into public health. It's for everybody. Don't say that you're in the academia, you're in the hospital. No, anywhere you find yourself, have that privilege and responsibility as pharmacists to educate anyone around us on this important topic, immunization, and if possible, get the certification. On this note, I want to appreciate all our members who are here with us, and so far so good. I want to thank God for His faithfulness throughout February 2025 till this day. Today marks the end of our clinical meeting for 2025. So we thank God for everything, for making us to still hold this clinical meeting, learning in order to improve our patient outcomes. I want to appreciate all of us. Thank you so much. We are going to enter into celebration mood, Christmas and New Year celebration. So we're going to start again in 2027, in 2026, sorry, on the 7th of February. And that topic on that day, we all love it. So thank you so much. Enjoy your Christmas, enjoy your holidays. See you again on the 7th of February 2026. God bless you all.
Thank you.
Thank you, ma'am. So as we draw to a close, I'd like us to listen to a write-up here for us to ponder on in the main chat. Someone sent a message that's saying, what are we doing about the custody of vaccines in our hospitals? We need to ponder on this and take action nationally. So I think it's important for us to actually ponder on that as we close. So like the chairman has said, we appreciate our presenter and we appreciate all our professors and pharmacists present here. And may God bless us all. If can someone close us with a prayer, possible? Anybody at all?
Please don't forget to make these slides available for us.
Oh, okay.
Shall we pray?
Sure. It will come.
Okay. Father, we thank you for all that you have taught us tonight through our erudite professor. Thank you for your grace and mercies. We appreciate you. You are the author of knowledge. We give you praise. Our Father, we pray that we'll not just be learners, but we'll be doers in Jesus' name. And as we retire to our different beds, Lord, we ask that we have sweet sleep and we'll be better energized to do more for you. Thank you, Father. In Jesus' name we pray.
Amen. Dr. Bello,
Amen.
thank you so much. The pan
Amen.
Thank you
lecturer.
very much for
Can't wait to see
listening.
your name.
Amen.
Thank you so much, Dr. Bello.
Thank you, Majama.
Thank you.
Yeah,
Thank you.
good evening. Good
Thank
evening,
you,
ma'am.
Professor
My question was
Bello.
omitted.